Hello everyone, I'm Fatemeh Azad, PhD student from Artificial Creatures Lab under supervision of Dr. Bagheri at Sharif University of Technology. This presentation is about hardware implementation of coupling of astrocytes via gap junctions. So first of all, I'm gonna to talk about astrocytes, star-shaped glial cells in the brain and spinal cord. Then I'm gonna explain about the interaction between neuron and astrocyte, and then I'm going to focus on the dynamic model of Lee and Rinzel astrocytes. In the fourth part, gap junctionals communications between astrocytes will be explained, and after all, we will have results and discussion. So, as all of us know, neurons are the most important cells in our brain, central nervous system. On the other hand, astrocytes, also known as astroglia, are star-shaped glial cells in our brain as spinal cord. In humans' brain, a single astrocyte can interact with up to 2 million synapses at a time. In fact, astrocytes are linked to gap junctions and regulate nearby synapses. Astrocytes react to neural activity by producing transient increase in their calcium concentrations. However, they are not able to generate action potentials or sparks, but they contribute in neural information processing by releasing transmitters such as glutamate or ATP in the synaptic cleft. So astrocyte can encode and respond to stimuli via the intercellular calcium oscillations. Researchers have shown that in response to a stimulation, I mean neural stimulation, astrocytes are able to release transmitters and propagate calcium waves over long distances. The study of astrocytes has been started since 100 years ago, and then about 3 decades ago, the experimental observations support that the fundamental role of astrocytes in response to neural excitation and synaptic plasticity modulations. In fact, astrocytes encode information in amplitude or frequency of its calcium oscillation. So this slide shows the interaction between neuron and astrocyte, IF neuron, in fact, IF neuron model and the Lorentz model of astrocyte with 2AG signaling. The 2AG signaling is indicated with the black line as an action potential arrives at the IF neuron. 2AG is released into the synaptic cleft, which results in release of IP3 into the astrocyte generating calcium oscillations. Then IP3 will trigger the calcium generation process, and finally calcium releases from the calcium store, in fact. So astrocytes, depending on the neural stimulation, intensity will react to the external stimuli. So here, these are the equations that shows the dynamic model of astrocytes. So as you can see, when a neuron spikes, we have this equation for neuron. In fact, the IF neuron is one of the most common neuron models used in computational neuroscience, whose equation is as follows. Rm is the membrane resistance, Tm is the time constant, V is the membrane voltage, and I sin is the input current from synapse. We assume that when the IF neuron fires, 2Ag diffuses into the synaptic cleft. From this equation, Ag is the quantity of 2Ag, tau Ag is the decay rate of 2Ag, Rag is the 2Ag generation rate, and TSB is the firing time of the IF neuron. IP3 is produced based on this equation. The production of IP3 is influenced by the amount of propagated 2Ag. The production of IP3 within the astrocyte is modeled as this equation, I mean dIP3, this equation in fact. So RIP3 is the IP3 production rate. IP3 star is the baseline of IP3 when the cell receives no input and it is in a steady state, in fact. Tau IP3 is the IP3 decay rate. Calcium generation is a function of three main fluxes, I mean J channel, J leak, and J pump, where H is the fraction of activated IP3. The expression for the fluxes are given by these three equations. As you can see, these three equations are based on calcium, IP3, and H parameters. So here we can see the gap junctional communications between astrocytes. There are two equations which describe gap junctional coupling of astrocytes. In these two equations, we have P calcium and P IP3 parameters, which are respective values for calcium and IP3 permeability at gap junctions. In this picture, we can see the gap junctional communication between adjacent astrocyte by glutamate release from adjacent neuron. Since the permeability of gap junction to IP3 is much higher than the calcium permeability, we use the equation number 2 to model the coupling of astrocytes. So in this figure, you can see the results of our simulation, our network, I mean one neuron and one astrocyte, to show the basic modulation in our astrocyte, in fact, AM modulation, FM modulation, and both modes. The following simulations were performed using MATLAB simulator. The basic AM and FM behavior of the model is shown in figure 1 B, C, D. In this experiment, the amplitude of a stimulus, I mean IP3, is set at 0.4, then it is increased to 0.5 in the time interval 306 seconds, and finally it is reduced to its original value. The second row in figure 1 shows the results of AM mode with the values of KAER equals to 0.1. Depending on the value of KAER, calcium dynamics switch from AM to FM mode and vice versa. The result of FM mode with KAER equal to 0.1 and RL equals to 0.05 is shown in the last row. In figure 2, we can see the results of R3 modulation during the IP3 stimulus signal with randomly incremented amplitude. Second row shows the result of AM modulation. The third row illustrates the result of FM mode, and the result of AFM modulation is presented in the last row. So here, this figure depicts the multimodal information encoding in the digital circuit. In fact, we first implemented our biophysical model in MATLAB simulator, and then we proposed a novel digital circuit for this biophysical model. So our digital circuit can encode the information, can encode the behavior and mimic the behavior. As you can see, first we apply the IP3 signal, and then, as seen in the second row, the amplitude of calcium oscillation increases or decreases as IP3 increases or decreases, whereas their frequency is constant. I mean, the second row. Column A depicts the result of biophysical astrocyte model simulated in MATLAB. Column B shows the result of linear model simulated in MATLAB. Column C represents the simulation of proposed digital circuits, and the column D illustrates the photo of the oscilloscope screen when the digital circuits are running on the Z board. As you can see, these results of MATLAB simulation are in good agreement with the results obtained in Vivada simulation for our proposed digital circuit. So this figure shows the physical setup for hardware testing of the proposed digital circuit. In this case, the digital circuit is run on the Z board, and the output signals after conversion to analog signals will be shown on oscilloscope. For analog to digital conversion, a 10-bit ADC was used. A 16-bit DAC was utilized to convert the digital outputs of the Z-board to analog circuits to be displayed on the oscilloscope. So here we can see the phase plane analysis to compare the dynamic behavior of the biophysical model and the proposed digital circuit. The phase plane analysis, calcium based on H, calcium based on J channel, H based on J channel are depicted in this figure. As we can see, the dynamic behavior of the original model simulated in MATLAB, shown in column A, is similar to the trajectories in the linear model simulated in MATLAB in column B, and the proposed digital circuit which implemented in Vivado simulator, which is illustrated in column C. So here, the present study, in fact, our novelty of our paper is shown in this case is here, a new angle to analyze neuron astrocyte and the astrocytic implementation in hardware. In fact, this is the first neuromorphic hardware which mimic the gap junctional communication between astrocytes. As we can see, due to the presence of gap junction, astrocyte can calcium waves in one astrocyte can travel within the interconnected astrocyte network, and we can see the encoded information as the AM modulation, for example, in second row, FM modulation in C row, and AFM mode in fourth panel. This model of astrocyte is able to illustrate different modulation, encoding information about external stimuli, in fact, based on IP3 signal, different signal values, and it can participate. Astrocyte can be participate in information processing in synaptic transmission. So we can see here the results and discussion. As I can said before, the present study showed a new angle to analyze neuron astrocyte and then astrocytic implementation in hardware. In fact, this is the first neuromorphic hardware which mimic the gap junctional communications between astrocytes. The proposed digital circuit is able to mimic biophysical information processing and encoding in astrocytes by different modulation, amplitude modulation of calcium, frequency modulation, or most modes. So these are the references which we used in our recent study. So thank you for your attention. I'm ready to answer your question.
Thank you very much. I have time for a few questions. Yeah, Mr. Bahramin. Yeah, go ahead.
Thank you for your talk. I hope you can hear me. Okay.
Yeah.
Yes. What's the benefit? What's the computational benefit of adding astrocytes to our neuromorphic architecture? Does it because one of the most important things in neuromorphic computation is that adding any element can help us in computing or lower the power consumption. What do you think is the benefit of adding astrocytes to our neuromorphic architecture?
Okay. In fact, this new neuromorphic circuit of astrocyte is able to successfully demonstrate AM, FM, AFM calcium signaling due to the external stimuli from neurons. In fact, as we can see in recent researches, astrocyte has a critical role in self-repairing paradigm. Self-repairing process. For example, astrocyte can participate in neurotransmitter transmission in synapses and has a critical role in different brain network. In fact, for example, as I studied before, it can have a good role in a self-repair process.
So you think it can help in learning algorithms, for example?
Yes, yes, yes. We see the new chip, I mean True North chip,
Yes.
that they use the many astrocytes in their neural network, spiky neural network, to mimic the biophysical behavior of our brain.
Very interesting. Thank you.
You're welcome.